Molecular cloning. The molecular cloning is the basic tool of gene manipulation और इसको मुख्तलिफ स्टेप्स में डिवाइड किया जा सकता है. Now, what are the different steps of gene cloning or molecular cloning? The first step of gene cloning is the isolation and fragmentation of source DNA. Now, what is the source DNA? Source DNA, it may be the whole genomic DNA from any organism from where you want to isolate your gene of interest, or even this may be the DNA that has been synthesized in vitro from the RNA, or even it may be the PCR amplified product of the DNA, and even we can use DNA that is in vitro synthesized. So, the source DNA may be of any type, and if the source DNA is the genomic DNA, then it can be fragmented. Fragmented का मतलब है कि इसको काटा जा सकता है into different segments. इसको हम कैसे fragment कर सकते हैं? We can fragment it by using different type of restriction enzymes, especially type 2 endonucleases that can cut the source DNA at specific point and can generate number of DNA fragments that may also contain your gene of interest. Now, you have digested, you have fragmented your gene of interest. Now, it is ready to be inserted into a cloning vector. Now, what is a cloning vector? Cloning vector, it is a genetic element that has the ability to replicate independently because of its own origin of replication. So, we can go to the next step, that is joining the DNA fragments to a cloning vector with DNA ligase. Okay. So, here we can cut the vector DNA with the same restriction enzyme that we have previously used for DNA digestion. कि हमें वही enzyme use करना है जिससे हमने अपना source DNA जो है उसको cut किया है. So that in most of the cases, depending upon the restriction enzyme, we can get the cohesive end. Now, cohesive ends, they are the single-stranded DNA segment of the double helix of the DNA that can again bind if complementary base pairs are available. Otherwise, in some cases, a blunt end they are also produced that can be joined or ligated by applying different techniques. Now, we have inserted our gene of interest into the vector, and then the next step is the introduction and the maintenance of the host organism. कि हम अपना जो recombinant vector है that contained our gene of interest, we will introduce it to a suitable host organism. तो जो सिंपल सोर्स है सुटेबल होस्ट ऑर्गेनिज्म का, इट मे बी द बैक्टीरियम, कि कोई बैक्टीरियम हो सकता है लाइक इकोलाई हो सकता है, बैसिलस हो सकता है. सो इट विल मल्टीप्लाई एंड इट कंटेन वैरायटी ऑफ recombinant plasmid, and some cells will also contain our gene of interest. Now, how we can screen our gene of interest are the transformants from thousands of microbes that constitute a gene library or the DNA library. Why? Because it contains number of transformants with our gene of interest or even some other fragment from the source organism or the source DNA. So, next step will be the detection and purification of the desired clone. We will select those transformants that contained our gene of interest. How we will select? We will discuss in the next diagram. And once we selected, then we can produce number of copies of that clone to get our desirable end product. Now, what strategy we will use to carry out this whole procedure, that is the gene cloning? First step is that is the source DNA. Source may be of any time, and here suppose it is the genomic DNA that is labeled here as the foreign DNA. It will be fragmented by using restriction enzyme, and then after its digestion into different fragments, then the vector, which is suitable cloning vehicle, which may be a plasmid, it is also digested with same restriction enzyme that we used for foreign DNA, and here it produces sticky ends, that is single strands of DNA with double helix. And next step is the ligation here, and once it is ligated, then it is ready for the next step, that is introduction of recombinant vector into a host. How it will be clear in next diagram. So you will understand in a much better way in this diagram the DNA cloning in a plasmid vector permits amplification of DNA fragment. So suppose here is our gene of interest. Here is our gene of interest, and here is the plasmid vector that we can use it as a cloning vehicle, and it contains selectable markers. Here it is the ampicillin, that it contains genes that can provide a selectable advantage for the microbes to grow in ampicillin medium, which is an antibiotic. Now in the next step, we will ligate our gene of interest into a plasmid vector that contains our gene of interest. And here, as I mentioned you, that it contains very important marker, which is the ampicillin. And onward, we will transfer this recombinant vector into the host organism, which is a bacterium. So when bacterium will grow on the medium that is supplemented with ampicillin, so only those transformants that contain our gene of interest, they will be able to grow there. In the next section, this plasmid, it will multiply into the host organism so that it will produce number of copies of the microbes. And then next laborious steps will start, that how we can clone or how we can find our desired clone from millions of cells by proper screening. So we will discuss this topic onward.